In Chapter 3, we're going to look at cells and tissues, and we'll start out by looking at cell structure. The objectives for this particular section are be able to list the three major parts of a typical cell and the function of each, list and briefly describe the function of the organelles of a general cell, describe the composition of the plasma membrane, and describe 4 specializations of the plasma membrane and explain their function. Cells, remember, are the smallest structural unit of all living things. In the organizational scheme, there's chemicals and then there's cells. So this is the next level up from chemicals. There are 3 basic components to a cell: the nucleus, the cytoplasm, and the plasma membrane, sometimes called the cell membrane. The nucleus is the control center of the cell. The cytoplasm is where all of the chemical reactions occur, and the plasma membrane makes the boundary of the cell. The nucleus is going to control all the cell activities. Typically, cells only have one nucleus. However, some cells have more than one, like skeletal muscle cells, and a few cells have no nucleus, like your red blood cells. The nucleus contains the DNA, and the DNA is the blueprint for the proteins. Cells respond the way they do because of the proteins they can manufacture. As we look at the nuclear structure, there are 3 major parts: the nuclear envelope or the nuclear membrane, the nucleoli, and the chromatin. The nuclear envelope is a double membrane. It has holes in it or pores in it that are pretty good sized. Inside is the watery part of the nucleus called the nucleoplasm. The nucleoli are structures inside the nucleus that are where the ribosomes are produced. Now, ribosomes are one of the organelles we'll talk about later. And chromatin is the DNA and some proteins that are associated with the DNA. DNA is a very slender thread-like molecule, and you've got a lot of it packed into your cells. In order to keep it from getting tangled up and damaged, it's packaged by these proteins. It's coiled up and wound up so that the chromosomes are shorter and thicker. The DNA is shorter and thicker, and it's not as likely to get tangled. Now, the DNA and the packaging proteins make up what are usually called the chromosomes. And chromosomes and chromatin are not quite the same thing, but they're usually used interchangeably. Now, this is a picture of showing the nucleus of the cell primarily. The nuclear envelope is this double membrane structure, and the pores, the holes in it, allow fairly large molecules to come and go from the nucleus. Here is one nucleolus. A plural would be nucleoli, and they're showing one of the organelles of the cell because of the way it is attached to the nuclear membrane. Inside here would be that nucleoplasm. The cell membrane, or the plasma membrane, is what we call a fluid mosaic model. Fluid because it's changeable. It moves. Mosaic because things come and go on its surface. It's not always the same thing. The basic structure of the cell membrane is that of phospholipid bilayer. A phospholipid, you'll remember, is a specialized lipid. It's got a glycerol, 2 fatty acids, and a phosphate group attached to it. And this makes it a rather unique molecule. The phosphate group is water soluble, but the fatty acid tails are not water soluble. If you put phospholipids into water, they automatically line up in a bilayer with the tails, the water-phobic parts, pointed inward, and the heads, the water-philic parts, the part that likes dissolving in water, facing outward. And that's just how that happens. Now, in addition to the phospholipids, there are some other things associated with the cell membrane. About 5% of the cell membrane is made up of glycolipids. Glyco means sugar. So these are combination molecules that are part sugar, part lipid. And these are found on the outside of the cell, and they're part of what helps the cells stick to other cells and helps other cells recognize it. About 20% of the cell membrane is cholesterol. Now, cholesterol is a lipid, so it's going to be involved in those lipid tails. those fatty acid tails, and cholesterol gives some structure to the membrane. Then there are two types of proteins that are associated with the cell membrane. Integral proteins are embedded in the membrane and go all the way through from one side to the other. Peripheral proteins are attached loosely to the inner surface of the membrane, and they can kind of come and go. And then there are a bunch of glycoproteins, that is, protein sugar type molecules. And these are very sticky molecules. This makes up what's called the glycocalyx of the cell, which helps protect the cell a little bit and make it kind of sticky. So as we look at this picture, these little balls here are the phosphate heads, and here are the two fatty acid tails. And you can see how they line up, heads out, tails in. And this gives us a really unique situation where we've got some interaction with the watery part that's outside the cell and the watery part that's inside the cell, but this is a lipid barrier that helps keep things from coming and going very easily. We have the glycoproteins and the glycolipids that are attached to the surface of the cell. The glycoproteins and the glycolipids make up that glycocalyx. The integral proteins go all the way through or are deeply embedded in the cell. The peripheral proteins are much looser in terms of their attachment to the cell, on both the inside and the outside, so they tend to come and go quite a bit. And these little yellow things here are the cholesterol molecules. And since lipid likes lipid, they're all woven here in these phospholipid tails, and they give some strength and some stability to the membrane. There are four specializations of the plasma membrane: tight junctions, desmosomes, gap junctions, and microvilli, and each has a special function. Tight junctions are found when we have these interlocking proteins between cells that hold the cells together very tightly. You see them up here, the interlocking proteins. They make almost a Ziploc seal. And we find these kinds of junctions between cells whenever we need a waterproof barrier. Things can't get in between these cells if they've got tight junctions. An example of where we might see this would be in the digestive system, where we would have the digestive enzymes in the stomach and the intestine, and we wouldn't want them to be able to seep between the cells and get into the tissues underneath, where they would start digesting away the tissues. Desmosomes are particularly good at anchoring things and allowing them to have some mechanical stress. And a desmosome, you've got these protein plaques on either side of the cell, and then you've got these linker proteins that come out and attach to each other. Now this holds the cells together, but there's more space between them, so things could go between the cells. But it gives you some twisting kind of activity, and the cells still stay locked together. We see this kind of junction between, say, cells on your skin, because your skin has to be pretty flexible. It has to move when you do. And if we've got these desmosomes between the epithelial cells of the epidermis of the skin, then as you move, you don't crack open. The third thing is the gap junctions. Now, gap junctions are actually little protein tubes so that the cells are connected and the cytoplasm in one cell can flow through to the cytoplasm of the other cell. So these cells are sort of connected and interchange material pretty easily. Whenever there is an electrical disturbance in one cell, that is immediately transmitted to the cell to which it's attached through this gap junction. So we see gap junctions in cells where we need rapid cell-to-cell communication. For example, the muscle cells of your heart, we only stimulate a few of those muscle cells to contract, and because they're connected by gap junctions, every other cell in the heart gets the message at the same time, so that the heart muscle contracts as one unit, not as a bunch of individual cells all contracting independently. And the fourth specialization of the cell membrane are these microvilli. Now normally cells are together side by side, and this surface that's on the outside is called the apical surface, and we only see microvilli on that surface. But these little finger-like projections give a lot more surface area to the cell. So whenever we have cells that are acting as an absorptive kind of cell, we have these microvilli on them. So instead of just having this flat surface to absorb across, there's all of this surface, a whole lot more surface to absorb through. We see these cells in particular in the part of the digestive tract that is absorbing nutrients because it allows for a lot more absorption of nutrients. The cytoplasm is all of that material that's outside the nucleus but inside the plasma membrane. The cytosol is the fluid that suspends all of the other elements. And the cytosol is mostly water, but there's dissolved elements and minerals and proteins in there. Then we have the organelles. These are the metabolic machinery, and we're going to talk about these in just a minute. And there will be some inclusions. Inclusions will be stored nutrients and various cell products. For example, your skin cells make melanin, and melanin is stored in your cells as inclusions. The organelles are the little organs of the cells. These are the things that carry out activities just like your organs carry out activities. And the first one we'll talk about is the mitochondria. The mitochondria are sometimes called the powerhouse of the cell. This is because this is where ATP is produced. Now, the only form of energy your cells can use is in the form of ATP. The organelle itself has a double membrane, so there's an outer membrane and an inner membrane. And most of the activity goes on in here, but this space in between the two membranes is very important in helping generate the ATP. Mitochondria are really interesting. They have their own DNA and their own RNA, and it's believed that at one time this inner part was a free-living organism that was ingested by a larger cell, and this is the membrane that the cell put around it when it ingested it, but it didn't destroy it, it didn't kill it. Rather, it sort of made it its little energy slave, and it makes energy. Because mitochondria have their own DNA and RNA, mitochondria can divide independently of cell division, so that if cells start having a larger need for energy, more mitochondria will be found in those cells to generate that greater amount of energy. The ribosomes are the site of protein synthesis. Some of them are free in the cytoplasm, and others are bound to a membrane. And the membrane they're bound to is this endoplasmic reticulum. The endoplasmic reticulum is sort of a labyrinth. It's a series of membranes and tubules that go all through the cytoplasm. This is like the circulatory system of the cell. Things can get into the little tubules of the endoplasmic reticulum, travel to a different part of the cell, and hop out someplace else. Now, I know you think that, gosh, a cell is such a small place. Why would you want to transport things around it? But remember, molecules are much, 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 much smaller than a cell. So to a molecule, the cell is a really big place. In addition, the endoplasmic reticulum sort of divides the cytoplasm of the cell up into little separate lab rooms. There's all kinds of chemical reactions going on in the cytoplasm of the cell, and some of them would cancel each other out if they happen side by side, but they can happen very close together in the cell if a membrane separates them. And all of the reactions in your cell are mediated by enzymes. And most enzymes have to be attached to a membrane if they're going to be functional. So the endoplasmic reticulum acts as a really big area for enzymes to attach, sort of a work table for the enzymes, so that chemical reactions can occur. Now, we said that the bound ribosomes were sometimes attached to a membrane. When they are, they're called the rough endoplasmic reticulum. And the rough endoplasmic reticulum, because the ribosomes are attached and the ribosomes make protein, are found in cells where a lot of proteins are produced. Smooth endoplasmic reticulum will not have ribosomes attached, and the kind of activity that tends to go on at smooth endoplasmic reticulum is the formation of lipids. So cells that need to produce lipids will have more smooth endoplasmic reticulum. The Golgi apparatus is a series of flattened membrane sacs. They're always closely associated with the rough endoplasmic reticulum because they take the protein that's made by the ribosomes that's attached to the endoplasmic reticulum and they process it. They package it up for leaving the cell because most of these proteins are going to be secreted by the cell. Mucus, for example, is a complex protein. It's protein and some other stuff. And while the basic protein is made at the rough ER, as the material passes between packaging plants, little flattened sacs of the Golgi apparatus, it gets its secondary and tertiary folding, it gets carbohydrates attached to it, and then it can be secreted by the cell. Now, some of the little secretory vesicles that come off the Golgi apparatus actually stay in the cell as organelles, like the lysosomes. These are some of those secretory vesicles where the material inside is not secreted by the cell but rather retained by the cell. And the material that's in the lysosome are the digestive enzymes. So the lysosomes are like the digestive system of the cell. If the cell is damaged in any way, the lysosome membrane can become very fragile and it can rupture. And when that happens, those digestive enzymes are released into the cell and the cell digests itself. This is called autolysis and sometimes is referred to as cell suicide, but it's not like the cell just gets depressed and kills itself. The cell has been injured, and by eliminating itself, it keeps from injuring other nearby cells. So it's a protective mechanism. Now this is our generalized cell. These are pretty much all the parts you can find in any cell. You see the nucleus here. We've talked about the nuclear envelope, the nucleolus, the nucleoplasm. This is that endoplasmic reticulum. And see how the membrane of the nucleus seems to come off and make all of these little tubules, this little maze-like structure. Now these structures that are attached here to the endoplasmic reticulum would be the ribosomes, so this would be the rough endoplasmic reticulum. And this is that Golgi apparatus, so that as proteins are made, they go into the Golgi apparatus and they pass from sac to sac to sac until finally the protein is all mature and just what it needs to be. And most of them, the secretory vesicles, move to the edge of the cell and release the contents outside the cell, that is, they secrete contents. But some of them remain inside the cell as lysosomes. Here would be your mitochondria. Here would simply be a vesicle. A vesicle is just a little membrane-bound structure. Here are some free ribosomes over here. Now, different cells will have different numbers and types of organelles. Like I said earlier, cells that, say, make a lot of mucus would have a lot of rough endoplasmic reticulum and Golgi apparatus. But cells that made something like the hormone, a steroid hormone, which is a lipid hormone, they would have a lot of smooth endoplasmic reticulum and not a whole lot of rough endoplasmic reticulum. Muscle cells, because of all of the energy they have to generate, have lots and lots and lots of mitochondria. Skin cells, not so many mitochondria. So it just varies as to how much of any one organelle is in a cell based on the cell's function. Now, in addition to the organelles, there's a special organelle called a peroxisome. These are sacs of enzymes like oxidases or catalases, and their responsibility is to detoxify substances. We say that they neutralize free radicals. Now, free radicals are molecules that can suck electrons away from other molecules and damage them. So some people believe that free radicals are what causes aging of the body. You make free radicals through cell metabolism. You don't really mean to make them, but you make a few every once in a while just because that's the nature of chemistry. And it's the peroxisomes that take care of these so that they don't do a lot of damage to your cells. These organelles, like mitochondria, are self-replicating. That is, if a cell is exposed to a lot of toxins or is manufacturing a lot of free radicals, the peroxisomes will increase in number to take on that increased activity. The cytoskeleton is also part of the cell, and there are three elements to the cytoskeleton: the microtubules, the microfilaments, and the intermediate filaments. Now the microtubules are hollow structures, like their name implies, and they're the largest of the three elements of the cytoskeleton. They're made of these little proteins, these little bead-like proteins, and they are constantly changing. You pull off some beads off one end, you add them to the other end, you make it longer, you make it shorter. Because these are so actively changing their length, these are involved in cell shape. They also keep the organelles in position. The nucleus, for example, is pretty much always found in the center of the cell. Why? Because the cytoskeleton holds it there. The microfilaments are primarily made of the proteins actin and myosin. And these proteins are capable of sliding over each other, so they're involved in cell motility and in shape changes of the cell. You'll talk quite a bit about actin and myosin when you talk about muscle cells. And the intermediate fibers look a little bit like a rope, one of those real heavy-duty ropes with lots of strands, because that's kind of what they are. And like those heavy-duty ropes, they're very tough, they're very stable, and they resist forces. These are the kind of filaments that anchor those plaques inside the cell for the desmosomes. Centrioles are organelles that are found near the nucleus of the cell. They're sometimes called the microtubule organizational center, or the MTOC, and that's because they're involved in making any of the microtubules, including the cytoskeleton microtubules. We typically notice the centrioles in the cell during cell division when they make something called the mitotic spindles, which are microtubules that help separate the chromosomes. They also form the basis for flagella and cilia. Now, flagella and cilia are organelles that allow the cell to move in some way. They both have the same structure. They're microtubules in what's called the 9+2 form. There's 2 microtubules in the center, and then 9 pair of microtubules arranged in a circle around that center too. Here they've sliced across one, and this is what it looks like under the electron microscope. The difference between the two is cilia are shorter, and they tend to cover a cell's surface. They tend to move stuff across the surface of the cell. For example, you've got ciliated cells in your trachea that helps move mucus up out of your lungs. Flagella are longer and typically fewer in number, like only 1 or 2. The sperm, for example, has a flagellum. So, taking one more look at our generalized cell with all of its parts, we've got microvilli here showing the ribosomes, the Golgi apparatus, your plasma membrane, nucleus, nuclear envelope, chromatin, nucleolus, all of those parts of the cell. Here's the centrioles showing up for the first time. All these things are mitochondria. And remember, different numbers of organelles and different types of organelles will reflect what the cell activity is. In summary, cells have 3 basic parts: nucleus, cytoplasm, cell membrane. They have numerous organelles that allow cells to carry out all the basic life processes: digestion, making energy, metabolism, all kinds of processes that they have to do. Cells will have different numbers of organelles and different types of organelles depending on their function. So while we've talked about a generalized cell that has all of the organelles in it, depending on a cell's function, it will have more or less of certain organelles. And the cell membranes are phospholipid bilayers. They have proteins embedded in them, on them, and through them, and some other structures associated with them. Okay, let's see if you can do this now. The part of the cell that controls its activities is the nucleus, cytoplasm, plasma membrane, or mitochondria. That was too easy. You knew it was the nucleus right off the bat, right? A tight junction would be found between cells that needed to provide for mechanical stress, increased absorption, a waterproof barrier, or rapid cell-to-cell communication. They're the Ziploc seals that give you the waterproof barrier. The desmosomes give you mechanical stress, the microvilli give you increased absorption, and the gap junctions give you that rapid cell-to-cell communication. The organelle that produces ATP, or energy, is the ribosome, mitochondria, endoplasmic reticulum, or the lysosome. Mitochondria is the powerhouse of the cell. That's what makes the ATP. Ribosomes are involved in protein synthesis. The endoplasmic reticulum is more like the circulatory system of the cells, and the lysosome contains the digestive enzymes. One more. The liver cells detoxify substances you ingest, like alcohol and drugs. In order to perform this function, liver cells would have numerous mitochondria, lysosomes, centrioles, or peroxisomes. The correct choice is peroxisomes. Remember, they're the ones that have those enzymes that detoxify things. Again, mitochondria, powerhouse of the cell, lysosomes, digestive system, and the centrioles are involved in the production of microtubules.